everyone. So in this lecture, we are going to be looking at plant development. But first, we are going to review. So in last lecture, we looked at nutrients. And we said that there are 17 essential nutrients that are needed by our plants for their healthy growth and development. 3 of them, carbon, hydrogen, and oxygen, they get from the air and water. And that comprises about 95% of the dry weight of our plants. The remaining nutrients, 14 of them, they get from the soil, and those are referred to as mineral nutrients. Now, we can break our mineral nutrients down, into macronutrients and micronutrients. And macronutrients are needed in the largest quantities, micronutrients needed in smaller amounts. And if we can further break down macronutrients into primary and secondary, with again, primary needing, our plants need primary macronutrients in greater quantities. And there are 3 big ones of our primary macronutrients. There are 3 of them: nitrogen, phosphorus, and potassium. And those are the 3. Most important isn't quite the right word, but those are the 3 that we need the most of in terms of quantity. Right? And I don't want to say most important because all of these nutrients are needed, right? You need all of them for the proper growth and development of your plant. That's referred to as law of the minimum, right? But in terms of quantity, we need those primary macronutrients the most. Now, we also said there's some other factors that are important in terms of nutrients, and one of which is element mobility. And that is the ability for these nutrients to be translocated to other areas of the plant, right? So to take those nutrients from one part of the plant and move them to another part of the plant. And this becomes really important when we start looking at deficiencies or nutrient deficiencies, where you will see nutrient deficiencies of your mobile elements appear in your lower leaves first. That is because those lower leaves are the older leaves. So as your plant is producing new growth, it needs nutrients to produce that new growth. If it's not getting them from the soil, it can take those nutrients from its existing growth from those lower leaves and put them into the new ones. Thus, you will see a deficiency first in those older leaves. This is as opposed to non-mobile elements such as iron, as a good example, where you will see that deficiency first in the upper leaves or the new tissue, right? Because the plant isn't able to take the existing iron in its other tissues and utilize it. It has to take whatever iron is available in the soil, and if it's deficient, it won't have enough. Right, so that's where mobility plays an important role, at least in terms of identifying plant deficiencies, nutrient deficiencies. Now, how our mineral nutrients get into our plant is via active transport, right? This requires energy. They're able to pump these mineral ions into the plant through the root hairs using energy in the form of the ATP. Now, we then broke down those 3 primary macronutrients. We started with nitrogen, and that's needed the most, or the greatest quantity is needed by our plants, and thus it's the most common deficiency. We then looked at rhizobia, which form that symbiotic relationship with our legumes, where they're able to fix nitrogen from the air. They can take it from the air and turn it into a form that is used by our plants. We also briefly talked about the Haber-Bosch process, which is the process where we are able to fix nitrogen from the air, combine it with methane to make a liquid ammonia that could then be made into plant fertilizer or combined with plant fertilizer. So this is the way where we're able to make chemical or synthetic fertilizers. It was revolutionary for farming. We will talk about the effects when we start talking about sustainability later on in this course. When it comes to a nitrogen deficiency, what we see is a yellowing of our leaves, and we're going to see that deficiency first on our lower leaves because it's a mobile element. We then talked about phosphorus. We said that's needed in the second most, right? It's you need nitrogen, most nitrogen, or need nitrogen in the largest quantities. You need phosphorus in the second largest amount. Now, mycorrhizae help in obtaining phosphorus to our plant roots, and just like our root knot bacteria or nitrogen fixing bacteria, our mycorrhizae are fungi that form a symbiotic relationship with our plants, where our plants give them a little bit of carbohydrates, and they in turn help the plants access these nutrients that they otherwise couldn't access, including phosphorus. Now, phosphorus deficiency shows up in our lower leaves, and our leaves will start to turn purple, right? It's pretty distinctive when you see it. And lastly, we looked at potassium. We said potassium is really important for that stomata opening and closing, those pores that regulate gas exchange and transpiration. And we'll start to see potassium as a yellowing of our leaf margins. And when we talked about deficiencies, we said that the important thing is that many of them can look similar. Thus, it's often best that you take a soil test, and you can get them relatively inexpensively now. You can test your soil as well as your plant tissues to really know what's going on. Because sometimes it may not be a deficiency in terms of the amount of nutrients in your soil, but it could be a root issue or a water issue or something like that that is affecting their uptake. We also looked at soil pH, and we said soil pH is important because it affects nutrient absorption, right? Some nutrients are more soluble or able to be absorbed better at different pH levels. And actually, if you go beyond certain pH levels, you can get into toxicity, nutrient toxicity, or some other toxicity. In general, a pH between 6 and 7 is ideal for most plants, right? Slightly acidic, because that's going to be in the best range to allow for the greatest absorption of nutrients. And the last thing we looked at was fertilizer. And we said there's different options with fertilizer, your synthetic fertilizers versus your organic. Your organic often need to be broken down either by weathering or biological activity, right? Bacteria in the soil, etc. You also have liquid versus slow release. And we said that on your fertilizers, you will often see an NPK number, right? And you may not see NPK, but those three numbers stand for the different amounts or percentage by weight of nitrogen, phosphorus, and potassium in your fertilizer. And common ones are 10-10-10, 20-20-20, 1-2-3, etc. There's a whole bunch depending on the type of fertilizer. And we talked about fertilizer rate, right? So how much fertilizer should we be applying to our plants? And that's going to depend on whether we are applying fertilizer with every watering versus maybe once every week or every 2 weeks, right? And fertilizer rates are written in terms of parts per million of nitrogen, right? Again, nitrogen is kind of the one that's needed the most. So we write our fertilizer rates in terms of parts per million per nitrogen. And in terms of rates, if we are watering or fertilizing daily, a rate of around 50 to 100 ppm is common, maybe a little bit more. If we are going to fertilize every 2 weeks, something in the range of 300 ppm is more common. And how we actually calculate this or create a fertilizer then is what we can utilize that ppm number, and we utilize that NPK number to know how much fertilizer we should add to water to create our liquid fertilizer, right? So if we are shooting for a 120 ppm rate, right, that's 120 ppm of nitrogen. Say we have a bag of fertilizer, water-soluble fertilizer that is 10-10-10. Thus, we know 10% by weight of that bag is nitrogen. To calculate how much weight of that fertilizer we should add, what we do is we know that ppm can also be written as milligrams per liter. So we have 100, we want to get 120 milligrams per liter of nitrogen in our fertilizer. That 10 stands for the weight, percent by weight. So we take 120 and we divide it by 10% or 0.1, and that is going to give us 1,200 milligrams per liter. That is how much of the fertilizer we need to add per liter to get a rate of 120 ppm. So 120 milligrams is equal to 1.2 grams. So for every liter of fertilizer, we need to add 1.2 grams of our water-soluble fertilizer, right? And that is how you would calculate your fertilizer rate. So that is that. Let us get on with the lecture. Now, we've talked about plant development a little bit this semester already, and we've been talking about all those factors that affect plant growth and development. But now we're going to actually go through the stages of the life cycle of a plant. And remember, that's what plant development is, right? It's the progress of a plant through its life cycle. And we're probably going to move through some of these steps or stages fairly quickly because we've talked about them before, right? We've talked about seeds and germination. We've talked about flowering and the process of reproduction and fruit, etc. So we'll move through those fairly quickly, but we'll spend a little bit more time on some of the other stuff that maybe we haven't talked about, at least in depth yet. So let's start at the beginning, right? We have germination, and we've talked about germination before. Remember, we said that there are two kind of big patterns of germination, depending on whether it's a monocot, which has one seed leaf, versus a dicot, which has two. Our monocots are mostly our grasses, where our dicots are other types of plants. And we said, like we said, there's two patterns. So there's hypogeal germination, which means takes place below ground. Those cotyledons remain below ground. Those are typical of our grasses. And there's epigeal germination, which is typical of our dicots, and those cotyledons rise above ground. Now we know there are a few factors that are needed for germination. And we said that many plants, particularly our wild plants, not so much our agricultural plants anymore, but our wild plants, all those seeds, or many of them, start off as dormant, right? And it's a state of suspended animation. And there's different types of dormancy that we've talked about. Exogenous dormancy, which is some factor outside of the embryo, a lot of times a hard seed coat that is going to maintain dormancy. And that's different from endogenous or physiological dormancy, in which it's a factor within the embryo or an aspect of the embryo that's maintaining dormancy. And we briefly talked about the different ways to alleviate them, right? Exogenous dormancy would be some sort of scarification. So just maybe rubbing that seed coat or rubbing that seed on a piece of sandpaper to damage that seed coat. That's going to let water in to kickstart this process. Whereas physiological dormancy, it's that process to alleviate that, we would do stratification, where we keep our seeds at just above freezing in a moist environment for a period of time. Right? And we did a lab. We have our seeds currently in the fridge right now, going through that process of stratification. Now, once dormancy is alleviated, if dormancy even exists in our seeds, we know that there are three factors that are required for germination: water, oxygen, and temperature. Now, once our plant has germinated and now have a seedling or a young seedling, it's going to start going through its vegetative growth phase, right? And vegetative growth, we can look at that both in terms of the roots and the growth of the roots, as well as the growth of the shoots or the shoot system. Now we've talked about roots again. We've looked at some of this stuff already, and if we're talking about our roots, we know our roots have a number of functions. They anchor the plant to the soil, they absorb water and minerals, and they can also store nutrients. And we've seen that again if we're looking at difference between monocots and dicots. Our monocots tend to have more of a fibrous root system, where our dicots have a larger single taproot. And the thing I want to note additionally in this lecture is that as our shoot system and our root systems are growing, they maintain a balance, right? As you're producing, or as your plant is producing more shoots or more leaves, right? That is resulting in more photosynthesis and more transpiration. So there's going to be greater water needs, right? And as a result, that shoot system has to grow and expand as well, right? So as the shoot and the root systems are growing, they're keeping a balance so that they are able to adequately provide the water or energy needs for the entire plant. Now that brings us to our shoot growth, vegetative growth, right? And we can look at different types of growth. We could have determinate plants or determinate shoots versus indeterminate. Now with the determinant, after a certain period of vegetative growth, flower buds will form at the shoot terminals. And again, at the tips of our shoots are those apical meristems, right? And after a certain period of growing with our determinate plants, those buds are going to become reproductive. And we'll see that at the end of this lecture. Right, and then they become reproductive and form flowers. Whereas indeterminate shoots will continue to grow, and there'll be branches off, or shoots or stems that branch off that become reproductive. All right, so your determinate plants, an example would be your bush snap beans. Your indeterminates would be pole beans, grapevines, trees. Tomatoes, if you've grown tomatoes, you'll see that you can get both varieties, both indeterminate and determinate. All right, so something to be aware of. Now, we can also look at the shoot growth patterns, right? Now, our plants have three main patterns of growth. There are annuals, there are biennials, and there are perennials. So your annuals are herbaceous plants that complete their life cycle, right, going from seed to seed in one growing season, right? So they go through everything from germination to vegetative growth to phase change, which we're going to talk about, flowering, fruit and seed production, all occurs in one growing season. This is a lot of our plants, our vegetables, a lot of our flowers that we grow around here. We grow them as annuals, right? Corns, peas, watermelon, rice, most of your cereal grains, marigold, sunflowers, examples are all examples of annuals. Now, biennials are herbaceous plants that require two growing seasons to complete their life cycle. And I talked about biennials, if you guys remember when we were talking about the different types of plant tissues, and we said that there are apical meristems and then there's the subapical meristem. I said subapical meristems are important a lot, oftentimes in our biennials. And that pattern of growth with our biennials is that often the first year of growth, they will develop their root system, and then they will form a basal rosette of leaves, right? Very small shoot, and mostly a rosette of leaves that forms on the ground, and they're going to take in energy and photosynthesize and store nutrients in their root system. And then at the end of that growing season, often the top part, your shoot system, is going to die. And then they will overwinter and it'll trigger something called vernalization, which we'll talk about in a second. And then in the following growing season, the plant will regrow the shoot system. And at this time, it will become reproductive. It'll produce flowers and fruit and seed, etc. And again, and I mentioned that subapical meristem because during that time where it becomes reproductive, it sends off a tall shoot. It's the subapical meristems that are playing a large role in the quick expansion of that shoot. So the last pattern that we're going to look at is our perennial plants. Now, these can be either herbaceous plants or woody plants, and they are going to live for more than 2 years, right? Now, if we're looking at our herbaceous perennials, both the root and shoot system can remain alive indefinitely, or at least in terms of the lifespan of the plant. But often, at least where we're found in a more temperate region, that shoot system may be killed off each year by frost or other factors. And then shoot growth will resume each spring from latent buds or adventitious buds at the crown of the plant. So the crown is the point of the plant where the roots meet the stem right here. So think about asparagus, right? Each year it sends off new shoots that you can harvest, but those roots remain alive and dormant in the soil. And then each spring it sends out new shoots from this crown. Other examples of herbaceous perennials are chrysanthemums, potatoes, mint, and catnip. Now we also have our woody perennials, right? Typically in our woody perennials, both the shoot and the root system will remain alive indefinitely. Think of your trees or shrubs that you find around here. Now there may be some the deaths or deterioration, which we refer to as senescence of tissues, right? Think of the deciduous trees that their leaves die and fall each year, but by and large, the root and the shoot system is going to remain alive. And just like with your herbaceous perennials, that especially in your temperate regions that have an annual growing season, you'll see that the same with your woody perennials that will have more shoot growth during the springtime, which is the growing season in this area. And the magnitude of that growth each season can vary considerably based on the environment, right? Temperature, amount of water, again, all these things that we've looked at already that can affect plant growth. Now this brings us to phase change, right? We had our germination, we had our vegetative growth and different patterns of vegetative growth. And then at some point during that vegetative growth, there's going to be a transition into maturity, where your plant goes from a juvenile plant to a mature plant. And the distinction between them is that your mature plants are able to reproduce sexually. Very similar to animals, right? They go through puberty. This is the puberty of our plants, right? And our plants go from being unable to reproduce sexually to being able to reproduce sexually. And again, if we're talking about sexual reproduction in our plants, we are talking about the ability, or we are talking about flowering, right? Because that is the process of sexual reproduction. So phase change marks that transition. Now, there can also be other morphological and physiological changes that are occurring, right, that are unrelated specifically to just the formation of flowers or that we may see in the plant. A good example is the English ivy that you see here in this photo, right? It has distinct morphological differences, especially in leaf shape and growth pattern from when it is a juvenile plant to when it is an adult plant. Now, there's a practical importance of phase change in terms of plant propagation, and that is that for some plants, they will, and if we were trying to take cuttings, right, which is a form of propagation, cuttings will only root if the plant is in its juvenile stage. And this is more so for some of our tree species, including your oaks, your beeches, your pines, and your pears, right? So it's important that if we're trying to take cuttings of these plants, we want to do that with plants that are still juvenile. Now, the duration of the juvenile phase is going to depend on the species. And if you look on the table over there, you can see that it varies greatly, right? From 0 or 1 to 2 months all the way up to 30 to 40 years if you're looking at like a European beech tree. So there's a huge difference depending on the species and the length of the juvenile period of our plants. Now, plants aren't actually measuring time in terms of that, right? They're not measuring the passing of days. But what they think, and what they think is how plants are able to measure the time or know when the time is to switch over to mature plant and go through that phase change is the accumulation of sugar, right? It all goes back to that photosynthesis, right? Because what is photosynthesis? It's taking carbon dioxide from the air and turning it into sugar. It's turning it into a glucose molecule. And what we found, at least in a study that was done around 10 or 11 years ago now, is that there are transcription factors in play, right? Remember, our transcription factors are proteins that can turn genes on or off, right? They play a good, important role in regulating the growth and development of our plants, right? Very, very important. Well, there are a family of transcription factors, the SPL family, that will turn on genes that will result in the expression of adult traits, right? So essentially, this family of these SPL genes, transcription factors, will turn on genes that are going to transition our plant into an adult. But though the transcription or the transcription of those transcription factors, right, is controlled by microRNA. And those microRNA, these other types of transcription factors, repress those SPL family transcription factors. I know I'm saying that word a lot, but right, think of the microRNA is essentially turning off the SPL family. And because of that, the SPL family is not turning on the genes that are going to turn your plant into a mature plant. However, what we found is that sugar will repress the expression of that MIR156 or those microRNA transcription factors, right? So essentially, what you need for your plant to turn, at least genetically, to turn into a mature plant is the expression of a number of genes. And those genes are controlled by the SPL family transcription factors. But those SPL family transcription factors are kept off by these microRNA transcription factors. And then sugar is going to repress those microRNA transcription factors, right? So essentially decrease them, allowing the SPL family transcription factors to be expressed and thus allowing your plant to transition from juvenile to mature, right? And that sugar, that accumulation, that photosynthesis is one of the triggers for that to occur. Now, once we have a mature plant, right, what marks a mature plant is its ability to reproduce sexually. And that means the ability to flower. So when a plant becomes mature, a change can occur in its apical meristems, right, the tips of our shoots, that switch them from being vegetative to reproductive. Now, most agricultural plants are self-inducive, meaning that they initiate or form flowers after they reach a certain morphological maturity. Right? Now, people often use a calendar date to predict this, right? As we'll see, right? If you've planted any sort of plant and you look at the seed packet, it'll say this many days to maturity, right? You've probably all seen that. But research has shown that measuring the heat accumulated over time provides a more accurate estimate than counting calendar days, right? And this should be obvious to you, right? When we learned about temperature, right? We know that temperature is going to regulate metabolic processes, and we know that our plants are going to grow and develop faster with warmer temperatures up to a point, right? That 86 degrees Fahrenheit. So if we want to be accurate with our planting and our calendar, right, of knowing when we should expect different life stages of our plants or when we should expect, them to flower, what we can do is keep a record and calculate growing degree days. And this is a way of assigning a heat value to each day. And then those values are added together through subsequent days, and that's going to give an estimate of the amount of seasonal growth your plants have achieved. Now, there are two methods, the temperature averaging method, which is the easy way of doing it, and then there's the Baskerville-Ehman method, and there might be more nowadays, but that method number 2 is a little more complex. Now, you can actually just, with technology now, there's probably websites. I'm sure there's an app on your phone that will calculate this also for you. But we're going to look at that temperature averaging method so you get an idea of how this process works, at least a simplified version of it. So to calculate growing degree days, what you do is for each day, you look at the maximum temperature and the minimum temperature. You add them together, divide by 2, and then you subtract from that a base temperature, right? And the base temperature is the temperature below which development stops for that plant. And that can vary depending on the plant, right? So essentially, you're getting your average temperature for the day, and then you're dividing that by a base temperature. So let's, right? So if we've planted corn and we want to track its development using this growing degree day method, we know that the base temperature for corn is 50 degrees. That should be Fahrenheit, not Celsius. That's a typo. So below 50 degrees Fahrenheit, no development will occur, right, with your corn. Now, if we have an example of a day and the high was 83 and the low was 61, we want to calculate the growing degree days for that day, right? So what we would do is we would take our max temperature, 83, we would add it to our low temperature of 61 and divide by 2, and then we would subtract the base temperature from that. So if we actually do that in practice, we have 83 plus 61 is 144. We divide that by 2, which gives us 72. And then we subtract 50 from 72 because 50 is the base temperature, and that gives us 22 growing degree days. Now that might not mean a lot to you right now, but importantly, right, there is a lot of data, and we have charts that show you how many growing degrees it takes for different plants to reach different stages of development, right? So we've got 22 growing degrees day for here, and that might make more sense if you know that it's going to take on average corn around 1,000 growing degree days for flower induction, right? So we can keep track of these growing degree days, and it gives us a better estimate of when we should expect different stages of plant growth or plant development. Now, the reason why people have shifted to some of these more complex methods of calculating growing degree days is because the temperature averaging method will underestimate growing degree days when temperatures are still low, right? So either early in the season or late in the season. And why this is it's best explained with this example that we're going to go through, right? So say we have an early May day and the max temperature is 57 degrees Fahrenheit while the low is 42. So if we calculate our growing degree days, we would do the same thing. 57 plus 42 is 99. We divide that by 2, which gives us 49.5. And then we subtract 50 from that, which gives us negative 0.5, right? So if we have a negative number, it's equivalent to a 0. Right, so we would say there's no growing degree days during this time, so we wouldn't include that, right, in our accumulation. However, if we look at the actual temperature for the day, we know that at least for a portion of the day, the temperatures were above 50 degrees Fahrenheit. They were above that base temperature. So we do know that there was some accumulation or some growth or progress of the plant through its development on this day. It just wasn't a lot. And it's not included if we were using this temperature averaging method. So we could be off in our calculations a bit. Now, the importance of this, again, it may not be extreme, but if you have a lot of low temperature days, that could add up. And, we have these other advanced methods that are a little bit more accurate. So if you have it available on your phone, it's probably easier to just use that method, right, than the old school temperature averaging method. Now, a couple more notes on our growing degree days is that, as we've talked about, crops don't grow any faster at temperatures above 86 degrees Fahrenheit, right? So that's the max temperature that we're going to use in our calculations. So even if we had a very high, a temperature high that is above 86, we're going to use 86 as our max always, right? So, for example, if we had a day in which it was 92 degrees Fahrenheit out as a max, we're not going to use that 92. Instead, we're going to use our 86, right? Because that's the ceiling of our plant growth, or the speed of our plant growth. Now, again, as I mentioned, this comes in handy when we're able to look at charts like these that show um, on average, or the amount of growing degree days it takes for these different life stages of our plants to occur, right? And this is going to help you. This could help you with the timing of fertilizer application, right? Your plants may need more fertilizers or a specific type of fertilizer at different life stages, and thus it kind of helps from a management perspective to know, okay, this is when it's occurring, this is when we should have our fertilizers ready, or when we should be applying, or maybe they may need more water at this time if you're irrigating. It's also helpful point of pests, because insects grow and develop a lot in the same way, right? You can use growing degree days to track insect growth and development, just like you can do with plants. And that could be helpful then from the standpoint of targeting pests or pest control, right? Whether either targeting them with pesticides or other control measures. So maybe this is when this particular pest is going to be an issue, and this is maybe when you want to apply some sort of pest control measure. So that's all essentially calculating our plants that are self-inducive in terms of flowering, right? They're going to go through a certain, they're going to build up their growth, right? And then a certain period of time, they are going to become reproductive. There are other plants that we've talked about that will initiate flowering based on other factors, right? And one of those is the length of day. And we've already talked about this process when we looked at light, and it's called photoperiodism. And this is changes in the growth and development of a plant due to variations in daylight, right? And often we are talking about this in terms of flower. So we've said that there are short day plants that are going to flower when the day length is shorter than some critical day lengths. There are long day plants that flower when day length is longer or greater than some critical day lengths. And that critical day length is individual to each species, right? It's unique to each species, the actual timeline of that. And then we have our day neutral plants that don't respond to this at all, right? We've learned about this already, so I'm not going to go too in depth with it. But for your curiosity, here are a number of plants that respond to photoperiodism. Both are long day plants and short day plants, right? You can see a bunch of them. I want to see if lettuce is included in here, because that's what we're growing. I don't see it in this. But there are a whole host of other plants that respond to photoperiodism that you can see listed there. Now, so we have our self-inducive plants in terms of flowering. We have our plants that will flower in response to photoperiodism. And we also have our plants that flower in response to vernalization, or require this. So this is particularly true of our biennials, right, often. But our vernalization is that some plants require exposure to low temperatures for flower induction, right? So they need a period of cold to stimulate them to flower. Again, think about our biennials, right? Where they have their initial growth, and then shoot system is going to die off during after the first frost, right? Or as the days get colder, and then they're exposed to that cold period, and then that's going to trigger them for the next season to become reproductive. All right, so that is this process happening right here. Those plants need to be exposed to a period of low temperatures for them to start flowering. And there are a number of examples: winter wheat, Brussels sprouts, carrots, olive trees, broccoli, hyacinth, tulips, lilies, all require vernalization. Now you can force this, right? Just like we can stratify our seeds in a refrigerator nowadays. You can force vernalization by exposing plants to cold temperatures, right, artificially. right? And this is done by growers who sell tulips or hyacinths in the spring, right? They're going to vernalize the bulbs in coolers already beforehand and then plant. So we know we looked at these couple things that will trigger, that will do flower initiation, right? Or induction, right? Or we're going to induce flowering, either self-inducive, photoperiodism, or vernalization. What's actually happening with flower development? Well, our apical meristems are changing from vegetative to reproductive, right? And those apical meristems are the tips of our growing points, right? Now, once this process happens, it is not reversible. But if environmental conditions do change, that bud can be aborted, or that flower bud can be aborted, right? So this would be if there was maybe an extreme temperature change, or if you had a change in conditions with our short day or long day plants, right? All of a sudden, maybe we messed up we're growing them artificially and we mess up our lighting system and it changes everything around. It could cause your plants to abort the development of their flowers, and in this case, secondary or auxiliary buds will continue with vegetative growth until those environmental conditions change and are right for flowering again. So flowers, again, the whole purpose of our flowers is this process of sexual reproduction, and we've talked a little bit about this in terms of pollination, and when we looked at pollination and seed development, right? But all pollination is, it's the transfer of pollen from the anther to the stigma, right? And we said that the anther is the top part of our stamen right here, the male reproductive part. The stigma is the top part of the carpel or the pistil, right? And ultimately, the sperm cells and the pollen need to travel down the style into the ovary, into an ovule, and fertilize an egg cell to create a new embryo. Now, most of your fruit crops and your vegetables are insect pollinated, right? So, and you will see that there are adaptations for that. Your flowers that are insect pollinated will often be brightly colored. They will have nectaries to attract different insects to them. But you can also have plants that are wind pollinated, like your grasses, right? Your cereal grain crops, they are often wind pollinated. So they will generally have more inconspicuous flowers, right? Because they don't need to attract these pollinators. Now, bees, by and large, are the most important pollinators, but there are also a number of other ones. Think of butterflies, flies just in general as well. And there can also be some interesting pollinators, right? Hummingbirds, you might have seen. Bats are very important pollinators for a number of plant species, including mangoes, guava, and agave. So people who enjoy tequila, they are only able to enjoy tequila because that tequila plant, the agave plant, is pollinated by bats. So they should really be concerned about protecting bats. So another interesting pollinator could be a snail. So the graceful awl snail pollinates a particular plant in the morning glory family. It's the only known example of a snail that pollinates plants. It's a pretty interesting study that they did. This is also pollinated by bees as well, but when the bees can't pollinate it, when conditions aren't right, it's apparently more suited for the snail to come in and pollinate. It's pretty interesting. Now, some of our plants are self-fertile, which means they can be self-pollinated, right? So they can produce fruit and seed from their own pollen. So their own pollen can land on the stigma of their carpel, or they'll land on their stigma of their pistil, right? Carpel, pistil, kind of interchangeable, right? And they will be able to produce fruit and seeds from this. Other plants are sterile, self-incompatible. So they cannot set fruit or seed from their own pollen. They need pollen from a different individual in order to pollinate. And what can happen in the case of our self-sterile or self-incompatible plants is that if their own pollen does pollinate them, it can result in parthenocarpy, which is the process by which our plants will be able to recognize their own pollen, right? And it's genetically controlled. Those plants produce a protein that allows them to recognize themselves. And as a result, self-fertilized flowers will not develop seeds, right? So they become seedless. Now, this can be we can take advantage of this sometimes with some of our seedless varieties, such as like oranges or clementines, can be produced this way through parthenocarpy, where the are pollinated with their own seeds, but they won't produce, they may produce a fruit tissue, but they won't produce seeds in this process. Now, when we talk about fruit setting, right, if we have, or not doing this process, right, if we have normal, either our self-compatible seeds or self-incompatible seed or plants that have been fertilized with other types of pollen, we've talked about that fruit is the mature ovary plus accessory structures, right? So if you go back to that image of the flower and the female part, the carpel or the pistil, we said there was the stigma, which is kind of like the sticky pad at the top, the style, which is the kind of long tube, and at the bottom was the ovary, and within the ovary are the ovules, and that contains the egg cells, etc. Well, those ovules become the seeds, and that ovary develops into the fruit. Now, in many plants, only a small percentage of flowers are going to develop into fruits, and this is because many of your flowers don't get fertilized, right? And many of your fertilized flowers are aborted as well. And some of this has to do with resource allocation, right? Many plants don't have the resources in terms of energy or nutrients to develop all possible fruits. Thus, in fruit production, right, a chief problem is to obtain the optimal level of fruit set. Now, if we have too heavy of a set, that is going to result in small, poor quality fruit, right? Because our plant is limited in its nutrients and it has to divide those nutrients up amongst a whole bunch of fruit. Those fruit aren't going to be as big or maybe have as high as a sugar content if there was fewer of them that the plant had to divide resources up to, right? So too heavy is a problem, but also too low could be a problem as your plants, as it then can become unprofitable, right? You grow a tomato plant and you pick off flowers, so you only have one tomato that grows. That's ultimately not going to be profitable. So what happens in practice is usually that half or more of the fruits are removed at a very early stage. This is particularly in the nursery field, fruit nursery, by hand thinning or machine shaking or chemical sprays, right? And there is something referred to as the king's fruit, particularly with apples, where they may thin, if you have, look at that cluster of developing apples right here, they may thin this to just the biggest one, and that's going to be referred to as the king's fruit, and they're going to let that develop, right? Again, for this process related to this resource allocation. Now, fruit growth and development is impacted by a number of plant hormones. And we haven't gone through these hormones yet, but we have touched on them in other parts or in other lectures. So in terms of our fruit development, there are a number of hormones involved, including auxins, right? Make our cells divide and elongate our cells, gibberellins, and others. So we've seen actually when we looked at strawberries, when we started talking about fruit in past lectures, and we saw that auxins are produced by the achenes, actually technically the fruit of your strawberry, and they cause the receptacle to develop, which is the big fleshy part that we think of as the fruit, right? So you'd had your normal development right here, but if you remove those achenes, again, technically the fruit, you won't have the development of the receptacle, so you'll have this as your strawberry. But if you remove those achenes, those fruits, and you apply auxin, you will still have the development of your strawberry receptacle, right? So we know auxin's important, at least for strawberries in development. Gibberellins are also very, very important in terms of making bigger fruits. And in fact, all of your grapes that you buy have been sprayed with gibberellins, right? All of your grapes that you buy at your store have been sprayed to make them larger, right? And it results, right? Here on the left are those that have been sprayed. Here on the right are those that haven't, right? Much, much bigger. We also have climacteric fruits, which release ethylene as they ripen, and these include apples, bananas, tomatoes, and apricots. And that ethylene is like a positive feedback that it results in increased ripening, right? So as these are ripening, they start to produce ethylene, and ethylene triggers more ripening, right? If you've ever had bananas in like a paper bag, right, you'll see they'll, as they start ripening, it kind of speeds up and that all of them will ripen. It's the production of this ethylene. Now we can actually treat plants or fruits with ethylene from a harvesting standpoint to make them easier to harvest, or you can treat them with ethylene to um, ripen after we've harvested them. So think of, we do this with tomatoes, right? We may harvest our tomatoes when they are green and then artificially treat them with ethylene to ripen them to make them into your red tomatoes that you will then buy at the store. So this brings us to our last stage of plant development, and that is aging and senescence, and then ultimately death. Right, so different species have different lifespans, just like animals, right, with different lifespans. It could be months to even thousands of years. But ultimately, your plants are going to senesce and then die. And senescence is the irreversible deterioration of cells and tissues of an organism, right? So senescence can be related to the whole individual, or you can talk about senescence in terms of a particular organ, right? Like each fall, the leaves of our deciduous trees will senesce, they deteriorate and then die and drop off. Now, propagation can technically extend the life of a plant, and we can do that through cloning, right? So there's actually a black Corinth grape that is a clone that has been grown in Greece for thousands of years, right? So we continue to create new clones of this plant, and that is going to extend the lifespan of the plant if you think of each clone as a continuation of that individual. Now, the causes of senescence are usually inherent physiological changes in the plant, but it can also be caused by pathogens or environmental stress, right? We know during leaf senescence that DNA, RNA, proteins, chlorophyll, photosynthesis, starch, auxins, and gibberellins all decline, right? It's a general decline of everything. In your annuals, particularly your determinate annuals, there is a decline in photosynthesis following flowering. Excuse me. The flowering is, especially with your annuals, is going to be a big trigger ultimately for the death of your plant. And what is actually happening is your plant is taking its resources that it has used to grow up until this point, and then it puts all those resources into flowering and then in fruit development. And after that, it's really the goal, for lack of a better word, of that plant, right, is to reproduce, and it puts all that energy into reproduction, and then hopefully those fruits and seeds get dispersed, and new plants will arise, and that original plant will die, right? So we can actually postpone senescence in these plants by picking off those flower buds, right, before seeds form, right? Because we're going to try and postpone that plant putting its energy into its seeds and its fruit. But ultimately,